Hello student, welcome back to my channel. So today's topic of discussion is isolation of microbial pure culture. So please watch my video till the end and do subscribe my channel and please do not forget to like my video. So let's begin with the introduction part. First of all, I would like to tell you the pure culture is also known as exenic culture. Isolation of pure culture, why it is so important for any microbiologist? Because microorganisms are omnipresent in nature, means they are everywhere. They are always found as mixed culture where different species of microorganisms live together. By using different techniques, we can isolate the particular microorganism and culture them separately to obtain a pure culture. That's why pure culture is most important technique for the any microbiologist. What is the purpose of isolating pure culture? The purpose of isolating pure culture is to study, explore, and utilize the microorganism. It is necessary to obtain them in the form of pure culture. After obtaining pure culture, it is tested, verified, and characterized in order to confirm the origin of microbial species. Isolation of pure culture has application in various fields such as molecular biology, bioprocess technology, clinical studies, pathological and agriculture field, etc. So, next question is, who developed the concept of pure culture? The pure culture technique was developed by Robert Koch in 19th century. What are the different type of pure culture techniques? First is streak plate, second one is spread plate, third one is pour plate. These three techniques are the commonly used technique in any microbiological laboratory. Fourth one is capillary method. Fifth one is micro manipulator method. These two are the advanced technique to isolate pure culture. Stick plate technique. So for stick plate, these are the some basic requirement which we need. Inoculating loop, sterile media, burner, alcohol, 70% alcohol, sterile petri plate, and incubator to grow the microbial cell. What are the procedure? What is the process to isolate the pure culture using streak plate culture technique? First, we will clean the bench using 70% alcohol. Switch on the burner. Then we will pour the sterile media, autoclaved media into the sterile petri plate and let it solidify for 10 to 20 minutes. After media will get solidified, we will incinerate the loop or heat the loop till it get red heated. Later on, we will take the sample using the sterile loop and streak it on the desired culture plate or desired medium such as native agar, Mekong agar, blood agar. It depends on the microorganism. Whether your microorganism is fastidious in nature, means it has some special requirement to grow, or whether it can grow on basal medium like nutrient agar, etc. So we can streak the plate in any of the method, whether it is a quadrant streaking or simple streaking. Quadrant streaking is much better because it gives you a higher rate of pure culture isolation. After streaking, we will incubate the culture plate at 24 for the 24 to 40 hour at 37 degrees Celsius for bacteria. Second technique is spread plate technique. Requirement for spread plate technique is alcohol, nutrient media, sterile petri plate, spreader, burner, and incubator. This is the process of the spread plate technique. First, we will clean the working bench with 70% alcohol. Later, we switch on the burner. Then, take 0.1 ml of mixed culture with the help of pipette and inoculate it on the surface of the sterile nutrient media plate. Dip, dip the spreader in alcohol and sterile that spreader into the flame. Later on, we will allow it to cool and spread the inoculated culture using glass spreader, as you can see in this picture. After this spreading, we will incubate the culture plate at optimum temperature for 24 to 48 hours. So what are the advantages of spread plate technique? It is a simple method. In this method, only surface colonies are formed and microorganisms are not exposed to higher temperature. And what are the disadvantages of this method? Disadvantage of this method is we cannot isolate those microorganisms who needs below the 6% of oxygen, means microaerophilic organisms. We can isolate microaerophilic organism only using pore plating technique. Third technique is pore plate technique. These are the list of requirements: sterile media, petri plate, alcohol, burner, pipette, and incubator. This is the protocol for doing pore plating technique. First, we will clean the bench with the alcohol, 70% alcohol. Take 1 ml of mixed culture with the help of micropipette. Inoculated it in the sterile molten plate, or pour the inoculated molten agar on the sterile pelletry plate. First, we will pour the culture specimen 0.1 ml or 1 ml into the sterile petri plate, and over that we will pour our touchable autoclave media and swirl the plate so that culture suspension get mixed into the plate properly, and later on we will let it solidify for some minutes, for 10 to 20 minutes, and after solidification we will keep it in incubator for 24 to 48 hours at optimum temperature, and after that we will observe the colony formation beneath the media or under the media. In pore plating technique, only those microorganisms will grow which required only below the 6% of oxygen. What are the advantages of pore plating technique? It is suitable for microaerophilic microbes. And what are the disadvantages of pore plating technique? Microorganisms trapped beneath the surface of medium and surface as well as subsurface colonies are developed, which make the difficulties in counting the bacterial colony. It is a time-consuming method and microbes subjected to heat shock because liquid medium maintained at 45 degrees Celsius and it is unsuitable for psychrophiles. We will talk about single cell isolation methods. These methods are advanced methods to isolate the pure culture. First in this technique is capillary pipette method. In this technique, there are several small drops of suitably diluted culture medium are put on a sterile glass cover slip by a sterile pipette drawn to the capillary. One then examines each drop under the microscope until one finds such a drop which contains only one type of microorganism cell. As you can see in this picture, this is the glass cover slip containing several diluted culture drop containing microorganism cell. These cover slip will be examined under the microscope for the search of the such a drop which contain only single cell. Later on, after finding the suitable drop, we will collect that drop using capillary pipette and transfer that drop onto the culture plate. And later on, that cell in the drop will start multiplying. And through this method, we can isolate pure culture. Method which is lying under the advanced technique is micromanipulator method. Micromanipulator method have been built which permit one to pick up a single cell from a mixed culture. This instrument is used in conjunction with microscope to pick a single cell, particularly bacterial cell, from a hanging drop preparation. The micromanipulator has micrometer adjustment by means of which its micropipette can be moved right and left, forward and backward and upward and downward, as you can see in this picture. This is the micromanipulator containing micro needles or capillary needles which is conjuncted with the microscope. With this we can select the single drop containing single type of microbial cell. In this we use microscope to pick a single cell from the hanging drop preparation. In capillary method we do not use hanging drop, but in micromanipulator we use hanging drop. Hanging drop of diluted culture are placed on a special sterile cover slip by a micropipette. Now a hanging drop is searched for containing only a single type of microbial cell. This cell is drawn into the micropipette by gentle suction, then transferred to a large drop of sterile medium onto another sterile cover slip. When the number of cells increases in the drop is transferred to the culture tube containing a suitable medium. This yields a pure culture of the required microbial cell. What are the advantages and How micromanipulator method work? What are the disadvantages of the micromanipulator method? The micromanipulator method works by removing a single cell from a mixture of cells. It sucks the single cell into the micropipette and transfers the single cell to a large amount of sterile medium, where the cell will multiply, and later on we will transfer that drop into the large amount of media so that uh, all microorganisms can multiply, and we will get pure culture of microorganism. What is the advantage of micromanipulator method? First, it being reasonably sure that the culture comes from the single cell. Second is obtaining strains within the species. Third one is isolated aerobic or anaerobic species can be grown in pure culture or subject to this single cell PCR. That's why we need such a advanced technique of pure culture isolation. Please do subscribe my channel and please do like my video. Thank you.